Alright, here is a quick review over protein synthesis. Alright, there are two stages of protein synthesis. So what we have here is we have a strand of DNA. Okay, in DNA, each of your groups of 3 are called a triplet. Okay, so each 3 nucleotides are called a triplet. This is your strand of DNA. So in your nucleus, the first thing that happens with your DNA is it unwinds and then it unzips. Similar to what happens in DNA replication, but this has nothing to do with DNA replication. Alright, a quick reminder on DNA, C is going to base pair with G, A is going to base pair with T, C with G, so on and so forth. Okay, so those are going to be their corresponding base pairs. So the first thing that happens in transcription is that we have a strand of mRNA that is created from a strand of DNA. So unlike in DNA we have T, mRNA has no T, it actually has a U. So here you can see the A in DNA is actually matched up with a U in mRNA. So that's going to be your uracil. There is no T in mRNA. So C still base pairs with G, but A base pairs with U. T still base pairs with A, but again, there is no U in RNA. So this is transcription happening in the nucleus of the cell. Once mRNA is created, it's going to leave the nucleus and it's going to go find a ribosome. Alright, so here the mRNA left the nucleus, it found a ribosome. That's what these little green squiggles are on the board. That's going to begin the process of translation. Okay, but first what's going to happen is that the DNA is done. DNA is pretty lazy, it just opens and closes. RNA does all the work. Alright, so part 2 we're going to look at translation. Part 1 was transcription, it happened in the nucleus. Part 2 is translation. This is going to happen at the ribosome. Alright, so here we have our strand of mRNA. On your mRNA, every grouping of 3 mRNA is called a codon. This whole structure right here that you see highlighted in purple is an entire structure, is your tRNA. Very top, this little puffball up here is your amino acid. And then these bottom 3 nucleotides, those are called your anticodons. Your anticodons match up with your codons. We can actually determine what specific amino acid is being produced by using this codon chart over here in the top left-hand corner. What we will do is actually look at your 3 nucleotides. So we've got C, A, and G. So over here, we'll look at our C, which is here, A, which is going to be up here, and then we've got our G, which is over here. So we've got kind of like a multiplication table. We've got C, A, G. So this one, the first one here, it's a little bit different than what I have. It's GLO, and here they've got it listed as GLN, okay, or I've got GLU. But that's how that's going to work. Okay, the second one here, we've got A, scratch that, we've got U, A, C. So we're going to do U, A, and C. And we've got the TRY. The TRY represents the first 3 letters in that, that amino acid's name. Okay, so once your first tRNA comes down, that amino acid is going to be brought down with it. The second tRNA is going to come down. The anticodons are going to match up with the codons. This little amino acid is going to jump and attach to the next tRNA. Okay, so here you can see those two amino acids have attached to each other. Now we have our third tRNA here. Again, we can look and see which amino acid is being produced there. So we've got G, U, G. So we've got G, U, G comes over here. So VAL is going to be the specific amino acid that's produced. These two amino acids are going to jump, attach to that one. And eventually, as this process continues and goes on, we get a long chain of amino acids all attached through a polypeptide chain, and it creates a they're attached by a polypeptide bond, which creates a polypeptide chain or a protein. And that is protein synthesis.